Cool, thanks. Yep, I'm Kirsty. I'm a PhD student at the University of East Anglia. I'm funded by the British Ornithologists Union, and today I'll be talking to you about drivers of ocean movement patterns in Round Island petrels. So we're all here because we love seabirds. They're some of the world's greatest travellers, and we all know about the pole-to-pole migrations of Arctic terns, the tiny little storm petrels that migrate from the North Atlantic down to off the south coast of Africa, and the albatrosses that circumnavigate the globe. And when we look in the literature, we can see that there's a diverse range of non-breeding strategies within and between species, and these are just a few examples here. And we can see that this happens across the different oceans as well. And so Annette's already touched on this, so thanks Annette. We see a lot of repeatability in migratory strategies and in puffins. We've seen this figure already. You can see that year to year they tend to go to the same place. And also from a meta-analysis that I conducted, published in Journal of Animal Ecology, we can see that they're also quite repeatable in their migratory timings. So this leads us to wonder what might be influencing these individual strategies. So some people have seen sex differences, particularly in sexually dimorphic species. We had a really good talk yesterday on black-capped petrels, so light and dark morphs tend to go to different places, so potentially genetics are having an effect. And also environmental conditions. We've seen that wind's really important, particularly for prosolaria forms. So this takes me to Round Island for my PhD. Round Island's off the north coast of Mauritius in the Western Indian Ocean. And Round Island has a strongly seasonal environment, which can be defined by two broad seasons, the austral winter and the austral summer. And this is characterized by the semi-annual wind reversal in the northern Indian Ocean. And this is a shift in the trade winds, and it affects things like ocean productivity as well. And on Round Island, we have the Round Island petrel. These are surface nesting pterodromas. They breed all year round, but in varying numbers across the year. And from this graph, you can see that the number of eggs, which is the grey lines, and the sea surface temperature in red, how that fluctuates and varies across the year, so there's a lot of variation. And the important thing here is that means that different birds on the island at different times are going to be experiencing very different environmental conditions. So that's my first question, is whether or not this is influencing the migratory strategies that the different petrels undertake. And then perhaps the coolest thing, or I think is really cool, is that although I'm looking at petrels in the Indian Ocean, the population of petrels here is actually made up of a hybrid complex of at least 3 species of pterodromas. So we've got the Trinidad petrel in the Atlantic and the Kermadec and Herald petrel in the Pacific. And on Round Island, we get 2- and 3-way hybrids of these species. So this leads me to wonder, does the genotype of the birds influence where they end up on their migration? So what data do I have? 421 light-level geolocators have been deployed on adult Round Island petrels since 2009. This has provided us with 263 complete migration tracks from 196 different individuals. And we also have incomplete migration tracks for another 46 birds. And from this map, you can see that these birds are covering the entire Indian Ocean at the population scale. But when we look at individual birds, they're really consistent in where they go and also when they go there. So I'm telling you this because for the next part of my analysis, I just focus on the first migration that we have for each of the individual birds. And I find this quite cool after seeing Annette's talk, because her tropical species tended to do different things, different times of year in such a tropical, unpredictable environment. So to try and identify the different petrel migratory strategies that these birds undertake, we use something called a Bayesian mixtures analysis. And very simply, this involved overlaying a grid over our tracking area. And this was a 40 by 40 grid. Each grid square was about 200 by 200 kilometers to take into account the spatial inaccuracies that you get with geolocator data. And quite simply what this does, it groups individuals based on the similarities and differences in the whole track distribution and compares that to others in the Bayesian mixtures analysis. So how many points there are for each migration in each of the grid squares. And this has given us some quite nice results. So these are the two most common strategies we see in this system. So we have some birds that go east and then north a little to the Somali Basin, and others that go directly north to the Arabian Sea. Some of the slightly less common strategies, we have birds going all the way over to the Western Australian Basin, some staying in the Central Indian Ocean, and a few going up to the Bay of Bengal. And then a couple of strategies that we just have one or two individuals doing. But this has given us 9 different whole migration routes that we can try and look at what might be causing birds to go to these places. So before I get into looking at those spatial structures, I wanted to see if different petrel genotypes are on the island at different times. So this is where those incomplete migrations come in. So I use all individuals which I have departure data for and genetic data. And the genetic variable that I'm using is based on an estimated membership value to the Trinidad type cluster. So I'm not going to go into this into much detail, but simply this is a value from 0 to 1, 0 being not very Trinidad-like, 1 being very Trinidad-like. And I'm going to direct you to Kat if you want to know more about this, because this is based on her microsatellite genotyping work she did for her PhD on the petrels. But if we look at this plot, so in the centre of the plot in the lighter colour blue, we have the birds that have a lower estimated membership value, and the further away in the darker, they're more Trinidad-like, so the higher values. And you can see that across the year, there doesn't seem to be much temporal structuring in when birds are present on the island. And when we try and test for this statistically, I've split it into the austral winter and the austral summer to try and disentangle between the different environmental conditions in those two seasons, and there doesn't seem to be much difference between those, and neither when we include the transition periods between the monsoon seasons, because obviously the trade winds don't shift at the exact same time every year. And we've tried to look at genetics in a few different ways. So my supervisor, Malcolm Nichols, also looked at these heavier Kermadec type birds, and there doesn't seem to be any relationship between when these heavier birds are on the island and when they're not. So that allows me to go on to look at if environmental conditions or monsoon seasons or petrel genotype influences the migratory strategies of these petrels. So I'm just focusing on these two strategies for the time being, because these are the ones we have most data for, and because I only have a subset of petrels with the genotyping data. So the strategies where we didn't have many in the first place, now we have slightly less. But that's fine, so we can use some binomial GLMs to try and see what's causing birds to go in these places. So in these polar plots, we've got them split by the two different strategies. Each line represents the day in which an individual departs on its migration. The longer the line, the more individuals that are departing on that day. And you can see that for both these strategies, they tend to be going at all throughout the year, maybe slightly less so for the ones that go directly to the Arabian Sea, but there doesn't seem to be much relationship, and statistically there isn't any difference between the two monsoon seasons. And again, it doesn't make any difference if we include the transition periods or not. And then for the genotype data, so on this plot, the y-axis is the estimated membership value to the Trinidad type cluster, 0 at the bottom, 1 at the top. And just as a reminder, these are the birds that we find in the Atlantic Ocean. And for the first strategy, there seems to be a lot of variation in these birds' estimated membership values. But for the second ones that go to the north, to the Arabian Sea, they seem to be slightly more Trinidad type. But again, maybe with a little bit more data, we might be able to find something significant there. So yeah, just to wrap that up, we found that the Bayesian mixtures analysis was a really powerful tool to cluster petrel migration routes. And based on my knowledge of the system, I think it's done a really good job. We didn't find any sort of temporal structure in when petrel genotypes were present on the island, and that didn't make any difference with how we looked at the genetic and environmental variables. And we didn't find any sort of significant effect of genotype or environmental conditions on migratory strategies in our current data set. But either way, I still think it's really interesting to be trying to figure out why they have such a diverse range of strategies from this one population. So yeah, with that, I just want to say thanks to all my supervisors, collaborators, funders, and you guys for listening to me rattle on this morning. Thanks.
Thank you very much. That was fantastic, and it's nice to see such a range of techniques come together. Are there any questions in the audience? You made everyone think really hard. I'll start off then. You looked predominantly at the two main strategies, but if I had to push you, where do you think that the strategies that are less used might fit into this?
Yeah, I'm really not sure because we don't have a huge amount of genetic data. That's the limiting thing. So you probably saw from some of the graphs that we don't have much in the, let me get this the right way around, the austral winter period, and that's because the genotype data that we had from Kat's PhD was focused on the time on the island where there were most birds to get the most data, and it wasn't until later on in the year that they wanted to start seeing what different birds were doing. So yeah, I'm really not sure, and I'm hoping like we're going to get some more genetic data processed soon, or move on to like using some more fancy stuff that I didn't understand. But yeah.
No, that's brilliant. Thank you very much. Has anyone else got anything at all? Yes, one in the back, and then one over here.
Thanks, Kirsty. So lovely to hear about Round Island petrels. I was wondering whether there was any age data on the birds that you were looking at, the different migration strategies for, and whether there's an age factor in that, and have you tracked juveniles?
Yeah, so I haven't looked at the age stuff for this analysis. I did for the repeatability work that I did, but again, we just don't have many known aged individuals. So yeah, I haven't looked into that. But yeah, we have started putting geolocators on juvenile birds and starting to get that data back now. There's not something that I've looked at, but based on Annette's talk this morning, they do seem to be a bit more exploratory than the adult birds. Yeah, so it's going to be quite exciting to see how those birds develop over time if we can carry on tracking them across multiple years.
Great. I think we've got time for another one if we want. There was one over here.
Hi, I think I'm getting the impression that you used microsatellites because of the availability of pre-existing data. But I was curious about whether there was any other rationale behind the use of that type of genetic analysis as well.
Yeah, so my main focus is on the geolocator and tracking processing. So yeah, I used data that was already available. We based it on the Trinidad type just because they're the most common birds that you find on the island, so we're more likely to see any sort of variation there. But yeah, I just used what I had available to me in the time that I've had throughout my PhD, but we're hoping, yeah, to use like SNPs and stuff later down the line, I think. So.
Okay, great. I think we'll say thank you very much there then. That was brilliant. And yes? And I'd like to invite Nina Dehnhard to the stage. She's going to talk to us now about shags and some different colony effects there.
Yes, good morning, everybody. My name is Nina Dehnhard, and I work for Nina in Norway. And please apologize my croaky voice. I had COVID 2 weeks ago, but I'm negative now. So I'm talking about habitat predictability in shags. And we all know that anthropogenic pressures in the coastal zone are increasing, and that includes fishery, aquaculture, boat and ship traffic, and kelp harvesting, among others. There's therefore an increasing need for marine spatial planning and taking seabird foraging areas into account. The problem then is how to identify important foraging habitat for seabirds. Sure, we can put GPS trackers on birds, but shags and also many other seabird species are breeding in many, many scattered colonies along European coastline. And really, we cannot put GPS trackers on shags or other seabirds in every single colony to look at their detailed habitat use. And based on that, identify important foraging areas. So the possible solution is to use predictions based on existing data from one colony to predict around another colony. So here the first step would be to identify environmental habitat preferences for one or several colonies for which we have data, and then predict the foraging habitat around a colony X based on the existing tracking data and those habitat models. So this is called a cross-colony transferability. And this has been attempted with moderate to low success for pelagic seabirds, but it hasn't been attempted for coastal seabirds. So we've tried this for shags now. And in green on this map, you see all the different shag colonies spread across the Norwegian coastline. So really, we cannot track all of those. And the black triangles show our 5 study colonies, where we used GPS trackers, either agotus or path track loggers. And the widest spread is between Hornøya and Jarstein, which is more than 1,700 kilometers. So this is a huge spatial range. We have 10 years of tracking data from Sklinna, which is our best covered colony. And at Sklinna, we have tracked up to 60 individuals per breeding season. For the other 4 colonies, Hornøya, Røst, Runde, and Jarstein, we included 2 years of tracking data from about 10 individuals each, so they were less well covered. And in total, our data set comprised of 5,264 complete foraging trips. So statistics. First, the habitat preference models. So we used binomial GAMs. So we had the foraging locations as 1, and we had random locations as 0. So foraging locations were identified based on turning angle and speed, and I used the expectation maximization binary clustering approach, EMBC, and this was validated with TDR data that we had from some colonies and some individuals, and this worked really well. If you have questions about this approach, come and talk to me later. The random locations were selected within the available habitat around each colony. So for each colony, we took a circle around the colony, which with the radius being the maximum foraging range, and this differed between colonies. So at Røst, it was the smallest shags foraged within 9 kilometers of the colony. At Sklinna, birds go up to 40 kilometers away, so this available habitat was larger. And then we randomly selected locations within this available habitat, and both the foraging locations and the random locations were then intersected with environmental variables. And those ones I used were distance to colony, depth, seabed slope, kelp presence absence, sea surface temperature mean, sea surface temperature variance, sea surface height mean, and sea surface height variance. And those means and variances were the temporal means and variances over the breeding season. For each of the year we had data. And I haven't written this down, but year was included as fixed effect in each of these models. And then we used the forward stepwise model selection to find the best model for each colony. So based on this, we then had the habitat preference models. And then the next step is this across-colony transferability. So then we used the data from 1 or several colonies to train the GAMs to then predict the foraging probability around another colony. The fit was used the fit was assessed with model cross-validation, and then that was compared to the actual tracking data. I used 2 different approaches and compared the results. In the first approach, we used 1 individual colony and the data from this colony, and the best model, which might have included depth and kelp and sea surface height, for example. So this best model structure from this 1 colony and that data to predict the habitat at 1 other colony. In the second approach, we used the data from 4 colonies. I call this the all colonies approach, to predict the habitat at the 5th colony. And we did this with all possible combinations. So the training data set was more diverse for the all colonies approach, naturally, because more data from more different colonies, more individuals, more variable. But also for the all colonies approach, the model was simplified because we had to find one that matched all the colonies. So we only included the key factors, which turned out to be kelp depth and distance to colony. I don't want you to look at every single line and every single number in this table, obviously. These are the habitat preference models, and here you see for each colony the best model. The most important part of this is depth and distance to colony were the most important variables across all colonies, so they turned up in the best model for each colony. And the kelp presence was also really important at all colonies. It was retained in all the best models at all colonies, but it was less important at Hornøya than at the other 4. If you remember, Hornea is in the far north east in Finnmark, and the reason why kelp was less important there was probably there isn't much kelp there because kelp has been grazed down by sea urchins in the north of Norway, so there is more sandy bottom available than kelp. Here's the model response curve for depth, and maybe not surprisingly, I haven't really introduced my study species, but shags are mostly benthic diving birds, so not surprisingly, they stay in relatively shallow areas. They mostly dive down to about 25-30 meters, and that is reflected here. So on the y-axis, the higher the value, the better the habitat model, or the better the more likely the shag was foraging in this habitat. So the deeper it gets, the less likely shags were foraging there. There's a little bit of a peak here for Hornea birds at deeper depths. They may sometimes do some pelagic, they may sometimes forage in open water, maybe feed on swarming prey, but it's more an exception than the rule. So shallow waters are important for shags, not really a surprise. Distance to colony, also not really a surprise. Why go far if you can forage close? Most of the foraging happens within the first 10 kilometers around the colony. But again, Sklinna kicks out here a bit. Sklinna is a very small, it's an archipelago located quite far from the mainland. So either the shags forage really close to the colony, or they have to travel quite far to get to other shallow locations, and that explains why they go so far and why we see this peak here further away. Coming to the results for transferability across colonies. So this is the model cross-validation results. These values represent the area under the receiver curve, AUC, and 0 would be really poor, and 1 would be the highest it can get. So if it's a 0.7 and higher, this means the model fits quite well. If it's 0.9, it's really good. And on the left, we see the training data set, and right, we see the testing data set. So this is the individual approach, where we use, for example, here data from Hornea to predict where birds at Røst were foraging. And we see this wasn't too bad for Røst, for Sklinna, yeah. It varies quite a lot between colonies, basically. If we look at Sklinna once more, so this was the data set that had 10 years of data and a lot of different individuals. We see Sklinna is really good, actually, in predicting where birds at the other colonies are foraging. But then if we look at how other colonies predict where birds at Sklinna are foraging, that's not so good. Using the all-colony approach, so using data from 4 colonies to predict where birds at a 5th colony are foraging, this was much better. So here, in this case, birds from the data from Røst, Sklinna, Runde, and Jarstein predicted really well where birds at Hornea would be foraging, and this was actually the case for all colonies. We are all over 0.9, so it's a really good model fit. And keep in mind, with the all-colony approach, this was the simpler model based only on depth, distance to colony, and kelp. And it is, of course, the larger training data set. So we can conclude, whoops, something happened there with my box. Transferability was high with the larger training data set, and even the foraging areas around Sklinna could be predicted really well when using the data from the other 4 colonies. So now I'm going to just show you some of the maps with where the models predicted where birds would be foraging, and this is in color range from red being really good foraging habitat to blue being not the open sea but poor foraging habitat, and white is landmass, and this should be the 100-meter bathymetry, and white you see where the colony is located, and this is Hornea. So here you see the map of Norway. Hornea is located in the northeast, and the black dots here is, I hope you can see these actually, where we had where the EMBC predicted where the EMBC identified foraging locations, and we see that actually, yes, most of these foraging dots are in the area where the model predicted where birds would be going, but there is also good habitat further away which the birds didn't use because, well, why travel far, right? Here's the same forest. We see birds foraging mostly in these orange and red areas, some going on this outer area here. So of course the model doesn't catch every single individual equally well or every trip equally well. But overall it's doing quite well. Here we see Sclina. Those black dots are just everywhere because we have so many data. But the key foraging areas are captured, and here you see why they have to travel so far, because there is not that much available habitat in this area here. And here the same for Runde. There seems to be a really good foraging area located down here, but of course the birds are not going there because they are not traveling that far. I'm skipping Jarstein because I'm running out of time and come to my conclusions. So we managed to test transferability of fine-scale habitat models in shags, and it actually worked really well. We found that transferability was high when using data from multiple colonies and years, and this really shows the value of comprehensive data sets in modeling and predicting important habitats. We can predict important foraging habitat around shags along the Norwegian coast with a reasonably high level of accuracy without having to track in more colonies. And I cannot emphasize enough how much of a value this has for marine spatial planning and designing protected areas. We all know the UN sustainability goals. They say we should protect 30% of the marine areas of each country by 2030. Norway is currently, or has been as of 2020, at 3.5% of its marine zone. So Norway is really lagging behind other European countries there, and we have work to do. And with this, I'd like to say thanks so much to my co-authors, all field assistants, the shags, and the funders.
Thank you very much. That's a phenomenal amount of data over such a good distance. I think we've got time for a couple of questions. Straight in the back.
Thank you very much. That was a really good talk. I was interested by the increased power of predicting other colonies from more the long-term tracking, as well as tracking multiple colonies. And I wondered if you had any thoughts on what might be better to invest in, like a longer-term study of one colony or more time tracking more colonies.
That is a really good and a really difficult question, or a really good question, and I don't really have an answer to it. I was contemplating of looking into this more and basically testing whether taking only 2 years of Sclina, for example, would be equally good in predicting where the birds from the other colonies are foraging. I haven't done this. I think it would be really worth doing, maybe at the next Seabird conference.
Thank you.
Great, thanks. Any other questions at all? Okay, I'll definitely go for one then. Given you've got such a huge range of data, particularly in terms of latitude, I was very impressed with the agreement between colonies. Given a future sort of trending scenario with climate change, do you think this is something that could become a really useful tool in the future for that?
It depends, of course, on how climate change is affecting these habitats. And I've actually looked into this, and I couldn't really find anything on how climate change is impacting, for example, kelp forests, because kelp seems to be the key point in a lot of this. And of course, kelp forest only occurs in shallow habitats, so this is linked. If it's too deep, there won't be any kelp, and so on. So that's a bit of a question on how does the habitat really vary. I mean, sea surface temperature and sea surface height, it's maybe not so surprising that it didn't pop up. It's very important for shag. And also, of course, they vary a lot over the spatial gradient we worked. I mean, Honea is icy cold compared to Jarstein. And shags, of course, have a range on where they occur in terms of temperatures and so. So I think the key question here is how is climate change affecting the habitats and the prey availability, because in the end, all the seabirds don't eat temperature, they eat fish.
Perfect, thank you. That sounds like a hopefully positive answer then. I think we'll say thank you very much there then. And we'll introduce Sophie Bennett now, who's going to talk to us about some cool stuff with guillemots.
Hi everyone, I'm Sophie. I'm in the final year of my PhD at the UK Centre for Ecology and Hydrology and the University of Liverpool, and today I'm going to be talking to you about the occupancy of breeding sites in the non-breeding season in common guillemots. So across taxa we see numerous examples of individuals investing huge amounts of time in defence of breeding sites, and this is because they want to obtain or retain sites of higher quality. And this can be particularly intense in colonial species like seabirds, where not only you can have large numbers of conspecifics competing for these sites, but also large numbers of other species competing for the same area. Now, from this idea, the site defence hypothesis predicts that these higher quality breeding sites should be defended more than those of lower quality. And as a result of this, we see that these sites are occupied earlier and more often, again, in many examples across the breeding season. And this leads to great intra- and interspecific competition for these sites as individuals trying to vie with one another to have these fitness benefits that we also see arise from breeding at these sites, so earlier breeding and also more successful breeding. Now, the occupancy of breeding sites in the non-breeding season is exactly what it sounds like, is where this behaviour takes place outside of the core breeding period. And we see numerous examples of this, again, in lots of taxa, but this is a Seabird group conference, so here are some nice seabird examples. And just in these examples, we see great intra- and interspecific and seasonal variation in attendance patterns and also the number of individuals that undertake this, which can reach more than 50% of populations. However, unlike in the breeding season, we don't really understand why individuals are coming back at this time of year and what the consequences of this can have for fitness. And a key cause of this is often just due to logistical issues with these data not being collected throughout the entirety of the non-breeding period. And we've heard a bit about, in this conference, about how this is getting improved for the future through things like remote sensing techniques. But as a result of this, we don't really understand what the fitness consequences of this behaviour are, despite the fact that it can take over more than 2/3 of the annual cycle. And resolving these knowledge gaps was the focus of our study, where firstly I wanted to quantify this behaviour on a really high resolution of site level throughout the entirety of the non-breeding period, and then investigating the relationships between these occupancy patterns, site quality, and then future fitness in terms of both breeding timing and success. And we did this using data from common guillemots, breeding on the Isle of May, which again we've heard a lot about. And in this population, this behaviour has been observed over a number of years, and so it's a prime candidate to investigate these questions. Now, to collect these data, we used time-lapse photography. So we installed cameras at 2 study plots, and we did this over 3 consecutive non-breeding seasons. And these cameras took images every 15 minutes throughout the entirety of the non-breeding period, which was from the beginning of October until the end of March, when birds first come back, getting on their sites for breeding and preparing for laying. And from these images, we extracted 2 measures of occupancy. The first was the first date that each breeding site was occupied in each period, as well as how often they're occupied, which was the proportion of survey days. So after processing all of these images, we were finally able to look at what these annual patterns of occupancy actually look like. So on the y-axis here, we've got the average proportion of sites that are occupied on each day throughout the non-breeding period, and this is averaged across both of our study colonies and all 3 study years. And we see that sites first get occupied around mid-October to mid-November, and it reaches a peak of occupancy of around 50 to 60% of sites being occupied on each day. And this peak is interspersed by 2 periods of decreased occupancy, the first associated with a second molt that takes place in the species, and the second gap is when they had some periods of really bad weather and when birds just weren't attending the colony in the numbers that they used to. So after solving this first challenge, and we have all these data, we were then able to look at how these occupancy patterns relate to site quality. Now, in order to do this, we firstly had to find a measure of site quality we could use. And we decided to use this average historic breeding success as our measure of site quality, and this has been shown by ourselves and another paper by Hannah Coco et al. To be a comparative measure to using direct physical measures that constitute the site's quality. But in order to satisfy ourselves that this was an appropriate measure to be using again in the sites in our study, we retested this relationship over our 38-year data set and found that this measure of the average historic breeding success correlates really nicely with these physical characteristics, and so we were satisfied that this was a good measure to use. So then looking at how this related to our occupancy measures, and we see some really nice clear results. So sites that were occupied earlier and more often were higher quality. And this, I also tested, and haven't shown this here, that those sites that are higher quality were also occupied for longer on each day as well. So this is already just a really nice, clear example that site quality does have a strong relationship with these occupancy patterns, and it may well prove to be a key motivating factor for why these individuals are coming back, and then why they're investing so much time occupying these sites when they do. So then looking at how this relates to future fitness. So we used two measures I mentioned earlier, the first being breeding timing, which was the date that each egg was laid at a site, and then breeding success, whether the chick successfully fledged. So looking first at breeding timing. So here we did find, you can't really see the error bars here, but we did see a significant relationship. So sites that were occupied more often did have a significantly earlier lay date. But this effect was quite weak. So for each 24% increase in how often sites are occupied, lay dates were just 1 day earlier. So there is a relationship here, but it doesn't really have great biological significance. But when we look at breeding success, we see a much stronger relationship. So sites that first occupied earlier had much higher breeding success, and that at its peak for each day earlier that a site was first occupied, the probability of success increased by up to 10.5%. And this effect was strongest for those birds that came back earliest. So if you come back earliest, it's really good. If you come back earlier later on, it's still good, but it's not as good as getting back before anyone else does. So putting both of these results together, we see that this occupancy behaviour can have key benefits for fitness for these individuals in terms of both timing and success. But it won't have escaped lots of your attention that these results I've mentioned so far kind of feed into one another, and this is exactly what we looked into a bit next to see how these effects have their relative importance to one another. So we built a path analysis where we fed in all of the relationships that I've talked about here, as well as some others that we tested for completeness. And we tried to see whether the relationship with return date was, say, as strong as that we see between lay date and success, which is well established for lots of seabird species, or whether actually just the effect of site quality going to breeding success was all that was really happening here. And what we found is that the path that was most supported was this one that goes from site quality to return date and breeding success. And this is really quite striking because return date happened 7 months prior to breeding, unlike lay date, yet it was the greatest predictor of breeding success. And we discuss all of these results a lot more in our paper, which should hopefully be out very soon. But moving on to what I'm looking at currently is this question of if occupancy is so beneficial for individuals, then why don't they all do it, or why don't they all do it more? So I also have some individual-based data for occupancy, and this is derived from geolocators. And again, I'm happy to talk about the technique of this with anyone. And so each horizontal bar here represents an individual, and the purple is when, or black as it looks, when birds were absent, and yellow is when birds were present. And I won't talk about all the patterns we see in this because that could take a long time, but basically individuals vary a lot, not just in when they come back and how often, but when these periods of absence and presence occur and how long these are. So at the moment, I'm looking at a couple of different questions to try and answer this question and to try and tease apart whether maybe there are different strategies at play here or whether perhaps there are different constraints acting upon individuals. So the first question I'm looking at is whether occupancy affects an individual's energetic and time budgets. So we've heard again from a couple of people that flight costs are really high in auks. So one of the things we were wondering is that if individuals are having to spend more time commuting to and from the colony, does that have a cumulative toll? I mean, that they are having to expend more energy to keep themselves going, or does it affect their allocation for, say, other maintenance behaviours? And we're doing this using both geolocators and time depth recorders. I'm also looking at how occupancy relates to migratory strategies and say whether we have more resident individuals that attend much more often and earlier, and more migratory individuals that do this much less. And again, this is using just the geolocators. And then lastly, whether males and females have differing roles in this site occupancy. So in the breeding season, it's commonly found that generally males have greater roles in this occupancy behaviour, which may also explain some of the relationships we found in our path analysis as well. So just to talk you through some results from this first question, so looking at how this relates to energetic and time budgets. So just briefly, we found no difference in daily energetic expenditure on days with or without occupancy. However, when we look at the time budgets of individuals, we find that individuals spend less time foraging on days when they're present at the colony. So this is where some of that, there's only so many minutes in the day, and the minutes that they're spending at the colony, which can be up to around 10 to 11 hours when you get towards February and March, is quite a lot of it is coming out of that time they normally spend foraging. And the longer they spend at the colony, the greater this trade-off that occurs for individuals. And what I'm looking at the moment is whether individuals have different levels of trade-offs, and so may be differently impacted, and may have different abilities to be able to undertake this behaviour. But ultimately, at the moment, we don't really see that occupancy directly affects energetics, but it does seem to affect time budgets. So just to end with 3 takeaway points. So the first, obtaining high-quality breeding sites is likely to be a driver of this behaviour. You come back earlier and you return more often and you get a better site. And as a consequence of that, you have fitness advantages in terms of earlier and more successful breeding. And although we see great individual variation in these occupancy patterns, this doesn't really seem to explain what we see in terms of daily energy expenditure, but we do see that this may affect the time that individuals have for foraging. So with that, I would like to thank all of the people who have helped collect this camera data, as well as all of the historic breeding data that we used in this study, as well as my supervisors, Francis, John, and Kate, for all of their support, and my amazing co-authors, Sarah, Mike, and Mark. And I'll just leave a paper plug at the end. The one on the left is the paper I mentioned, where we looked at site quality, and we looked at this in relation to population size and trend. Like I say, I had hoped that this other paper would be out by now. It's not, but hopefully it will be next week. So thank you very much for your attention.
Thank you very much. That was really, really good. I wonder if we've got any questions?
Thanks, Sophie. I was intrigued, so you've got some information on individuals. Do you get squatters? Like, I want to upgrade my site. Do you have any insight into that from your data?
So because it's a good question, and for the occupancy data that I have, so we've only got 3 years, and the turnover isn't that great, but there was another really good paper by Hannah Coco that did look at involuntary and voluntary movements in relation to site quality. And yeah, you do generally see that individuals do try and move up if they can, but the time series we have here isn't long enough, but it's likely to be occurring, because if you could, you'd try and move in earlier, definitely.
Oh, one down here.
A lovely talk. I'm just reminded of Emily Shepherd's talk the other day about the importance of good landing sites. Do you think that might play any role in determining what constitutes a good quality site?
Yeah, so again, punching up someone else's paper, so Mike Harris and a few others looked at what the specific characteristics of sites that are beneficial for guillemots, and because on the Isle of May, most of the sites are on the west of the island, and I don't think there would be huge differences, say, in terms of like landing and takeoff, but things that were important were, say, like the slope of the site, so like the angle to the sea, the height above the sea, and also kind of like the number of neighbours, and kind of whether you're on a big ledge or whether you're on like a discrete platform. But yeah, so there's a number of different things, and I think in other colonies that would be a really cool thing to look at.
Brilliant. Anyone else? Oh, yeah, one more then.
Hi. I'm really sorry if I missed this, but I don't think I picked it up. I was wondering whether the birds you were looking at were colour-ringed, and if they are, whether you've been able to consider breeding experience from your long-term studies within this.
Yeah, so a proportion of these birds are colour-ringed. It's really difficult. We thought it'd be easier with the cameras we had to pick up the actual individual colourings in the pictures. We had to find alternative ways to do it, which is why I used the geolocated data. Unfortunately, most of the birds that are colour-ringed are in a new study plot that was set up for my PhD, where it started in 2017, so we don't have a lot of data. But again, that'd be a really cool thing to look at. And I think one thing I'm trying to tease apart is whether, what's the relative importance of the site quality and the individual quality, and a huge part of that, the individual quality, may also vary with age, and hopefully if we keep this going, then we'll be able to answer that question in years to come.
Okay, brilliant. Thank you very much. That was incredibly interesting. And for the next talk, we'll introduce Jack Thorley, who's going to take us back down into the southern hemisphere again to go and talk about some albatross.
So, hello, I'm Jack Thorley. I'm currently a postdoc at the University of Liverpool, and in today's talk I'm going to be presenting data from wandering albatrosses to pose the question whether personality can mediate the reproductive consequences of broad climate phenomena. And the lens through which I'm going to try and address this question is through movement behaviours, as is very obvious from this meeting, individual differences in movement behaviour are ubiquitous. So decisions about whether to move, when to move, and where to move are pervasive across seabirds. And often, the intrinsic factors related to individuals explain a lot of this variation, so sex, state, and age of individuals. But increasingly, evidence across the animal kingdom is suggesting that personality, so these other consistent individual differences in types of behaviour, such as boldness, activity, sociability, can also contribute to patterns of movement among individuals. And so perhaps the most commonly reported that you'll see is this positive correlation between boldness and exploratory tendencies. And so personality, by providing this link between, by affecting movement, can affect individuals utilise space, acquire resources, and access mates. And that for this reason, it can have profound, or potentially have profound ecological and evolutionary consequences, and personality itself might be a potent target for selection under changing climatic or anthropogenic forces. So against this backdrop, I'll introduce the wandering albatross. I'm not going to do too much work doing this because Lizzie did a wonderful job of introducing the wandering albatross yesterday. So all I really want to say is that the population, the data that I'm using comes from the Crozet Islands. So you can see South Africa to the north, Antarctica to the south. If you have really good eyesight, the Kerguelen Plateau to the right. And the challenge that's faced by these wandering albatrosses, like many seabirds, particularly during breeding, is how to navigate these vast areas of ocean efficiently in order to find these ephemeral patches of prey, so in the albatross's case, commonly squid. And obviously they're equipped with this sort of diverse sensory apparatus to help them do that, but also their morphology makes them extremely well adapted to do this with their high body mass, which gives them a high wing loading, large wings, and their use of dynamic soaring behaviour. And so these birds are incredibly wind reliant. And again, in that context, it's interesting because the winds in the Southern Ocean are actually changing, and this is most often related to the southern annular mode, which is a major climatic oscillator in the Southern Ocean. And so on the left, you can see that from the 1960s to the 2020s, I could have plotted this a bit further back, this is generally increasing in amplitude. And one of the consequences of this, of most relevance to the albatross, is that a positive southern annular mode is associated with a strengthening and a contraction of the westerly winds towards the higher latitudes. So in the graphs on the right, you'll see latitude on the y-axis, average wind speed or average zonal wind. And then the latitude of Crozet is shown by the dotted line. And in particular to the south of Crozet, where the winds are where the males are predominantly foraging, you see the strengthening from the 1980s to the 2020s. And for this reason, because the southern annular mode is so strongly related to wind and albatross reliance on the wind, the argument has been made that it's this southern annular mode that is actually driving increases in the population and the breeding success on the Crozet Islands. And in particular, I guess perhaps more tellingly, if you take the arithmetic mean, so you look at actually the within-year variation in the southern annular mode, compare it to breeding success on the right, you can see this the reasonable correlation. There's obviously a lot of scatter, but this is controlling for nothing other, like no density dependence or any other effects. And so using this sorts of information and other information from the system, which has been the focus of long-term study from the 1960s, led to Henri Weimerskirch to sort of develop this conceptual framework for how climate, foraging, and demography are linked in the wandering albatrosses. So individuals have some phenotype, which is sort of made up of genetic components and some aspects of the environment that affects their foraging performance in whatever trait you might think is salient or relevant. Foraging performance itself might be influenced by the climate directly, in the albatross case potentially through wind, but also the climate's effects on affecting the resource in the marine environment. Obviously, good foraging can result in increases in body mass, or perhaps more accurately, sort of differences in on-body and off-body capital that birds can then use to finance reproduction and survival. And so the purpose of this work is really to sort of incorporate boldness into this framework as one potentially important aspect of individual phenotype that can sort of mediate these links between climate, foraging, and reproduction. The data we've got. So how do we go about doing this? Well, we can sort of rework this framework by putting on sort of relevant environmental and foraging parameters. So in this case, we're looking at trip-level characteristics during incubation. So we can take wind speed and its quadratic, and look at how this affects both the proportion of search behaviour engaged in by a bird across a foraging trip using extractive computer marker models, and also the path distance. And these two effects together can sort of be mediated and affect ultimately trip duration, which we're expecting is sort of a salient fitness trait in that if you have really long trips, potentially that's detrimental. And so what this really is like a putative causal model that we'll then build personality onto afterwards. And so I won't go into too much details here, but the approach we can use is called structural equation modeling. And the real power of this approach is it allows you to incorporate these direct and indirect effects and potentially get a bit more mechanistic idea of what's driving, at least driving these links between environmental parameters, foraging, and whatever trait you're interested in, I guess. And the other thing to just state is that by doing this in like a Bayesian approach, it's this really nice way of like propagating error through the model. And then, yeah, we're hoping that it's breeding success. So very briefly, I'll touch on the data that we're using to do this. So boldness has been collected pretty extensively since 2009 and repeatedly across individuals. So it's the response of birds to human approach. Foraging data takes the form of incubation trips, and that's from GPS loggers. Predominantly I got used, sampling at sort of anything from 1 to 15-minute resolution. And you can already see here this already mentioned distribution where females tend to head to subtropical waters, males head more south towards the poles. And then we overlay on that reanalysis data from the wind and other environmental things I'm not really touching upon today, and other controlling variables. Very, very briefly, boldness, the way it's scored is this like increasing scale where individuals with very low scores are essentially shy and individuals that have high scores are bold. And this is essentially sort of a graded scale, and so then we can kind of turn this into a continuous distribution if we sort of make the assumption that this threshold trait is underlaid by a Gaussian distribution on the liability scale. So we fit mixed effects models, we can get the position of individuals along that axis with some degree of error, and that's what goes into the models. And suffice to say that boldness is both repeatable and relatively heritable. So in terms of the results, before we incorporate personality, the way you read these, it's a little bit maybe intimidating, but essentially all you need to know is that each arrow is a single path coefficient, a single directional relationship. Black lines represent positive relationships, red negative relationships, and dotted lines like weakly supported effects. It's usually easier if you just do the plots from this sort of thing. So in the top right, you'll see that for females, as wind speed increases, the proportion of time in search behavior goes up. Independently, as wind speed increases, path length goes down. And then also, bottom left, proportion of time in search behavior also leads to decreases in path length. So these things start to like play off against one another, so it's quite difficult to necessarily work it out. So then what you then do is then you propagate that, and then you say, ultimately, how does wind speed affect trip duration through these different routes? And then if you want to interrogate the reasons afterwards, you do so. Males, you see that there's something quite similar going on. Really, the only change when I flick between is in the top left panel, where in extremely low winds, males are either not really engaging as much in, or they're engaging a bit more in search behavior, but that slope only starts to pick up past in high wind speeds. So as I say, you can then sort of take all this information and predict across the causal network, and then you can see that for females, increases in wind speed lead to generally declines in trip duration, whereas for males, there's this interesting effect whereby when wind speeds are low, they're not really going on very long trips, and that's, I guess, potentially because they can't forage very effectively at that point, so it makes more sense for them to incubate and then maybe pass the buck on to the female to do the foraging at sea, maybe. So then the real interest of the talk is then, well, how does boldness sort of play into this framework? And so the way I've approached this is to include boldness as both a linear and a quadratic predictor on both the proportion of time invested in search behavior, the path distance, and the trip duration. And so when you do this, all you really need to take from this is that when you look at the boxes that can include boldness and boldness squared, most of those sort of are dashed, which means there's not really strong support for a biological effect of boldness on those aspects of foraging behavior. The potentially one interesting exception is in the males, the bottom left, where you see that birds that are bolder tend to spend an increasing proportion of time, reduced time in search behavior. When you look into that a bit more, that's because bold birds are prioritizing travel rather than search. They're quite exploratory in their tendencies. This is generally suggesting that boldness isn't necessarily particularly important in this causal framework, given the assumptions that I've sort of built into the model and the traits I've looked at. And this is confirmed sort of when we then sort of overlay this additive effect of boldness onto the effect of wind speed. So you can see that there's very little like predictability when you include boldness into this approach. And that's particularly relevant when you take into fact that when you look at the y-axis here, sort of 12, 14 days, you only really start to see downturns in reproductive success, the ability to hatch a chick beyond 15 days, which is when abandonment happens. It's not really reaching that threshold very often. But obviously one of the caveats here is that trip duration is quite a coarse metric. There's a lot going on in there. And also, I guess, the problem with albatrosses is they're so good at reproducing that the variance between a good year and a bad year is sort of 70 to 80%. So trying to make that link between coarse trip things like trip duration and fitness is quite challenging. And so one thing we could do is step back and say, okay, well, let's pretend that we don't have any foraging information on the birds, and we just use all the information we have on personality and all the information that we have on reproductive success. And we can just ask, like, how is selection playing out on those traits, on boldness occurring over time. And so when we do this, essentially what you're doing is for each year you're taking the relative fitness, did a bird breed, did it not, and standardizing the trait, boldness, and then you're just performing regressions with a bit of extra tweaks in there to control for like the nonlinearity. And so the way you look at these plots is at 0, with that 0 on the incept means there's no selection happening in that year, or there's no consistent directional selection. Above the line is selection favoring bold individuals, below the line is selection favoring shy individuals. And as you can see, like over the sort of from the 90s onwards, there is this steady trend towards selection favoring shy individuals over time, suggesting that maybe this causal model is like missing something, or there's some other aspect of the biology during the breeding season that we're missing, or even the pre-breeding period. And so the question is like, what's really driving that, and that will motivate more work. But the nice thing is that when selection is operating, it's very aligned in both males and females. And so that means that if we want an evolutionary response, which could be predicted given that it's heritable, that there isn't any antagonism that could possibly break that apart in the future. But in conclusion, so I can say that boldness has been correlated with various aspects of albatross foraging behaviour before, and been quite successful when looking at probably finer scale details of foraging behaviour. But the evidence that personality affects how individuals utilise the wind at trip level scales and how that affects foraging behaviour is more limited based on the data I'm showing. In general, there is selection on boldness acting via reproductive success more generally, but how that relates to foraging style or climate is currently unclear. But it's worth saying that perhaps boldness effects are much more pronounced in other parts of the life cycle, and like with colleagues in America, that's sort of like ongoing work, like building these much more comprehensive demographic models and looking at vital rates across the life cycle. And if anything, the real take-home is that I think that the incorporation of both indirect and direct effects of personality, or indeed any trait you're working on, and looking at how that affects fitness, is likely to prove beneficial in trying to work out the adaptive variation around that trait and the potential that it could evolve in all the threats that seabirds are facing. And with that, I'll thank you for your time. I'll thank my colleagues and collaborators, in particular Dr. Samantha Patrick, and I'll take any questions.
Thank you very much. There's some fantastic analysis in there, and I wonder if we've any questions to begin. At the back, and then we'll jump.
Thank you for a great talk. I wondered if you had any information or maybe intuition on whether boldness affects how they interact with threats, maybe like fishing vessels and stuff like that. Thank you.
Yeah, so Omri Weimerskirch is currently doing that. Essentially, there are boldness effects on the interaction with, I guess it's mostly tuna longline fisheries. But from memory, the effects are both age and sex dependent, so I can't really pick out. It's not a nice, easy story for me to tell you where bold birds go to the boats. There's something more subtle in there that's actually quite difficult to pick apart. So yes, that work is ongoing and is close to completion, but I can find out, but I don't know off the top of my head the exact details.
Okay, yeah, I think we've got one more, and then we'll move on after that.
Hi, I'm not a seabird specialist, and I was looking at your boldness scale, and I was wondering why is flying away and leaving the reproductive attempt for a while is the higher boldness status?
We get this a lot. So I guess the short answer is it gets validated through additional assays, so in particular the response to a novel object. But I guess more broadly then, when you start to relate it to these other aspects of albatross behaviour, which has been done before, it starts to align with the fact that this high response is boldness. So for example, there's links between boldness and like rates of divorce have recently been found, and essentially most of these results relate to bolder birds have like a faster pace of life, and so there's various traits and aspects of their life history which ties into the fact that individuals that are more responsive do that. But yeah, I take the point that other people might like to interpret that differently, or might even not call it boldness. They might prefer to call it something like coping style, like reactivity. And so there's an element of subjectivity about what we're labelling the trait for sure.
Okay, great. Thank you very much. We'll thank you again.
Okay, hi. So what I'm going to present is part of a pilot study we led in the University of Gdańsk, where we try to investigate vocal communication between breeding partners and parental care. So because it's a pilot study and you're probably already tired, I promise you no more than 3 graphs and a cute little birds at the end. So, communication and parental care. Why do we care? So as you may know, parental care is a widespread strategy among birds, and it's even more important in seabirds, given the harsh environment they have to live in. So because of that, parental care has been a really important topic, research topic in behavioral ecology. And thanks to that, we quite well know now why they do that and how they do that, how they do share the parental care. But it seems that only recently we started to look at how they achieve that, how they actually share the parental care. So this question started to be investigated in songbirds, where they started to see that specific calls or increasing calling rates between partners could predict the following incubation bout or just the turn taking. And more recently, some studies also started to look at that in seabirds, showing that partners that were vocalizing more tend to share more equally the work. But acoustic in seabirds, in this context, is still really unknown, and they have a great potential for this question. So this is why we started to think about that in seabirds. So we decided to start in Little Auks, and like many other seabirds, they're not just cute, but they have really interesting features. They are monogamous, they breed with the same partner for several years, they have 1 chick per year. And in this specific species and this specific study site, we also know quite well how they coordinate the parental care during chick rearing. So parents alternate their trips, so they coordinate their work. And like other seabirds, they are loud. They do vocalize together. And what is important and interesting is that they do it not only during the mating, but during the whole breeding season. So it seems that it's important not just for mating, but for the whole breeding thing. So to start to look at that, we went to the field in Hornsund colony in Svalbard. It's a well-studied colony, so we have birds that are ringed for many years. We went there in 2020 during mid-incubation. So we focused on mid-incubation because it would give us the best condition to study that. As pairs, individuals are already mated, but there is no chick yet, so it's easier to isolate the communication between the breeding partners and not with the chick. Despite COVID, we managed to follow 14 breeding pairs, for which both individuals were color ringed, molecularly sexed. So for all these pairs, we installed a camera in front of the nest in order to know the activity of the birds. So which individual was doing what, when, and for how long. We did that for a continuous recording of 60 hours. So why 60 hours? Because we know that during incubation in this colony, the foraging trip can take up to 30 hours, so we wanted to make sure to have at least 1 incubation or foraging trip for each of the partner. So in parallel to that, we also installed microphone inside of the nest to record the communication of the partner, but this was not done for the 60 hours because, again, it was COVID, there was only 2 persons in the field, so it was just not possible. But still, we managed to get some meetings with vocal interactions. So this is how it looks like, but I'm sure you would like to know how it sounds like. That can be much more louder, but this one is quite soft. So I won't go too much in details in the methods, because I don't want to kill you with that, but I would be happy to discuss that with you later if you want.
But basically, we were just listening for hours of that and trying to isolate the calls during those meetings to know how many calls they were making. So once we had all of that, the first step was to look at how they share the work during incubation, because we knew quite well about chick rearing, how they alternate the activity, but we didn't know how they share the work during incubation. And what we saw was that, first of all, the duration of incubation varies a lot. So here you have activity patterns of several nests, and those durations, they vary a lot between and within pairs. And this is not a question of sex. So males and females, they vary the same way. So yet, they do very different incubation bout duration, and yet, during these 60 hours, they manage to do the same amount of work. So here you can see the proportion of incubation that is made by the female. So we have one lazy female here, but in general, they do 50% of the work. So how they do that? We don't know. Well, we didn't know. We still don't know, but we try to know. So one of the first suggestions that was proposed in other studies when it comes to sharing the work was a trip-to-trip adjustment. So there were different possibilities, so either reciprocal, so I will do exactly the same that my partner did, or compensation, so if he did less, I will do a bit more, or I would do more if he did. I would do more if he did. I would do less if he did more. Well. But we didn't find any relationship between successive incubation bouts. So that doesn't solve the question here. And this is where acoustic kicks in. So instead of looking just at incubation bout, we started to look at what's happening in between when they meet, when they can communicate. And we did find a relationship here. With the time they spend meeting, a positive relationship. So the more they spend meeting, the longer will be the following incubation bout. But what is more interesting is that during this meeting, the more they spent vocalizing, so the number of calls they did, the longer will be the following incubation bout. Well, so yeah, the more they communicate, the longer the following bout. But I won't speculate on these results yet because they are really preliminary and we only have one meeting per nest, so it's about 15. But here my point is not that we don't know exactly what's happening, but there seems to be a link between the two. Acoustic is doing something here. So we need to investigate that a bit more with a bigger sample size, but the next step would also be to not look only at the number of calls, but the difference between those calls, because we show that there is a variability in that too, but we don't know yet enough about the repertoire of this species. And then also it would be interesting to set up experimental approach once we know more about these specific calls. And in the bigger picture, it would be interesting to link this acoustic communication with other information about foraging ground condition, body conditions, because we know from previous study that visual displays can serve as transmitting information about this information about foraging and body conditions. So my message here would be we know how to watch birds, seabirds, but we have to start listening to them because they have a lot to tell us. And we started with this visual display. Now we have to look at the acoustic display as well. And also how it will change through time. We have data for that, but I won't go too much into detail also. So from that, I would like to thank you and thank, of course, all the field assistants, master students, the Polish Polar Station, and all people involved in this project. That was a pilot study, so also no Twitter yet, and all suggestions are welcome and questions. Thank you.
Thank you very much. I think we can all agree that was fascinating. So I'll open it up if anyone has any questions.
It's lunchtime soon, so.
Thank you. That was a great talk, really interesting. I was just wondering, did you also record breeding success? Because that would be really nice to know, and if you're going back and that's part of the plan as well.
Yeah, we do have information about breeding success. I can't quite remember. 2020 was a difficult year, so I'm not sure we have it for all the pairs. Yeah, but the thing is, like, 2020 was difficult. 2021, we will have all the information. Problem is that it takes a year to analyze. So, yeah, in the future, we would like to know that, link the, how well they coordinate, how well they communicate, and the breeding success, yes,
Great.
for the future.
Thank you. Awesome.
I'm just going to buzz straight on in here as well, if that's okay. Was there any differences in between who was doing the vocalizing, or if you have any idea, in terms of returning birds or leaving birds, or even sex differences?
We would love to look at that. It's not possible now because the micro is inside the nest, and inside the nest we cannot see, and from the acoustic we don't know which one is starting, if it's the incoming birds or the birds that were incubating. One of my colleagues is trying to do something about individual signature, of course, but it's very complicated with this species. But in the future, yes, we would like to look at that, maybe having a camera inside, though we know they don't like having anything inside the nest. So it's, yeah, we would love to see that.
That's fair. Any other questions at all? Oh, sorry, I did.
Hi there. I was just wondering if you could speculate on any other roles of vocal communication in the species. Do they also communicate if they're the only one at the nest, or if there's a chick present? Just, yeah, other roles of communication.
Yeah, when the chick is present, it makes the whole story more complicated because then you have 3 individuals. There is, not sure I quite understand the question.
Yeah, no, I mean, often there's, you know, a relationship between chick calls, chick begging calls as well, and adult parental care. And I wasn't sure if in Little Auk that if the adults are sometimes there alone and not with their partner.
Yeah, there are. There is also, they vocalize when they're alone in the nest. And we don't know if they vocalize to the egg, because in zebra finches, so it's not seabird, but they do that. They vocalize to the egg, and some studies show that it was to prepare them for harsh environment. But in Little Auk, we don't know. They do vocalize all the time, and we don't know what exactly they're doing. So,
thanks.
That's great. Thank you so much. That was brilliant.
Thank you.
And finally, we will have Lewis Fisher-Reeves, who's going to talk to us about sort of the visual landmarks in navigation.
Yeah, definitely. Okay, so hey everyone. Thank you for staying till the bitter end. We're almost there. So my name is Lewis, and I'm going to be giving a talk on the potential use of visual landmarks during the homing of a critically endangered shearwater, the Alle shearwater. Firstly, just a quick thanks to my supervisors, Tim and Ollie, and to Tash, Sarah, Rhiannon, and Miguel for collaborating and helping with the data. Firstly, a quick introduction to me. I'm a first-year PhD student at the University of Oxford. I have a real interest in the uses of biologging data in animal behaviour, as I'm sure many of us share, and specifically solving the mystery of these really long-ranging movements that we see birds go on, and what a better model here than shearwaters that travel thousands of kilometres. So I studied balearic data in the winter before a long field season on Skoma, pictured here with me and Trina handling acute Manxie. So this is sort of a pilot study that I did pre-fieldwork. So as I sort of jumped into the field of navigation at the start of my PhD, I read these really fascinating theories that we've heard plenty about of long-ranging navigational cues that may be used, and that touches upon things like the magnetic inclination angles and the magnetic field, olfactory gradients and their use as map cues. and even inferring the position relative to the sun. But a lot of these theories are prefaced with their sort of viability over smaller scales, whether the information they present has enough resolution to provide animals and birds with homing information at smaller, more refined scales. And despite this potential for error, we see that birds get home time and time again. So there must be something getting them home, and I was hoping to figure out that today. So this got me thinking about how I would navigate. What would I do? Well, I'm sure as many of you can relate, if I was to hop in my car and, say, drive back to Oxford, I'd say, "Hey Siri, can you take me home?" And she would, and it would be great. She'd get me most of the way, but I'd reach a certain point where I'd start to recognize junctions and buildings, and I'd no longer need Siri's guidance because my familiar area would be informed enough that I could get home on my own. And I started thinking, well, why can't birds do this too? We've seen in experimental studies with pigeons that when they have exposure to familiar landmarks, they can get home better than without. So perhaps shearwaters can do this too in a natural context. There they are there. And this brings me to my study species, the Balearic shearwater. As you can see here with my hand model, Alle, my supervisor, they are really cute cave-dwelling seabirds. They forage down in the Balearic Sea and occasionally the Mediterranean Sea. The study population that we monitor are on the island of Mallorca in Spain, specifically in Cecilia Cave, which is the largest known breeding colony of these birds. Sadly, as I'm sure many of you know, Balearic shearwaters are critically endangered through many anthropogenic effects. And so a lot of work is being done into studying the movement and behavior of these birds. The great thing about that is that we have GPS data available, which means that we have really good natural insight into their navigational strategies. So this brings me to the data that I have available. We've done several seasons out there collecting data. Here's my supervisor, Tim, and the team down in the caves collecting some GPS. And from several years of tracking, I've got 19 suitable GPS tracks that I could use for this sort of pilot study. I noticed that on first glance of this, a lot of the birds forage to the north of their colony. If we see here, so the outbound track is the grey along the blue arrow, and then this homing trip is in the red. A lot of them forage on the southern coast of Spain. And it's important to note here, and I'll touch upon later, it appears that there are no land masses between their colony and this coastline. And this makes a really nice natural contrast for the potential use of landmarks in the study. So before I jump into some results, I want to take you through some of the, what I think are very cool methods that we use to study navigation. How do you go from a GPS track of a free-ranging bird that just shows movement to sort of getting insights into their navigational cognitive proficiency? So firstly, you need to identify homing. Now, I won't dive into this too much. It's a bit statsy, and it is the last talk. But we use something called backwards path length. Now, if path length is the cumulative distance that a bird travels, backward path length is that just reversed, with homing being sort of the point of home being the first point on the data. If we show that on a graph, again, don't worry too much, but essentially you can see that there are these really linear sections of data, and this correlates to directed movement, the latter of which would be the inbound flight and the first being outbound, but obviously it's the inbound in homing that we're interested in, which would be this section here. You can see a really nice state switch between this sort of non-directed foraging movement and then these really linear sections, which we'd identify as homing. We can then take some really cool metrics from this. Again, I think they're cool. I'll focus specifically here on things called homing efficiency and instantaneous deflection, which I'll run you through now quickly. So homing efficiency is essentially sort of a measure of straightness of travel along a beeline direction. And when we say beeline, we mean from any location that the bird is at currently, what is the quickest way that it can get home? And that's often a straight line between the bird and its colony. So if we use this example, that's definitely of a shearwater and not of a bad clip art of Microsoft. We can see that if the bird travels from point A to B of 100 meters, and in doing so reduces its distance to its colony from, say, 300 meters at point A to 250 meters at point B, we'd say that this bird in this movement had a homing efficiency of 0.5, as by moving 100 meters, it's reduced it by 50. And we'll use the same example for instantaneous deflection. This one's a bit less mathematical. It's simply the difference in direction of travel of the bird, and again, this optimum direction home. So here, if the bird traveled 40 degrees and the beeline from its original point was 90 degrees, it would have a deflection of, therefore, 50 degrees. So now I can bring you to some cool results that I did. Do these birds home? Well, we know this already, that yes, they do, but it's nice to show you some stats to prove that how clever these birds are. They do, in fact, home. So not only do these birds fly in non-random directions, but they also fly with a significant preference towards their homeward goal. This was done using Rayleigh tests, which I'm happy to explain over lunch if people would like. In the data that we had, I noticed that there were 3 key foraging areas, so A, B, and C on this map. Granted, the vast majority of the data is from point A, but we have a couple of birds going to these other two locations. Again, the grey lines are sort of the outbound trips and foraging, and then these red lines are their homing journeys. On the left, we've got a nice rose diagram, and each arrow here shows the mean direction of travel for one of my GPS tracks. So if we look specifically at the portion A at the bottom, we can see that all of these birds that forage north of their colony have a real strong preference to fly south on their homing trip. We should expect, right, because we know that these birds can home. But do they use landmarks to improve this homing? And obviously that is what I'm really interested in. So before I could analyse this, I needed to figure out, well, when are landmarks available and how do they use them? Obviously, you'd imagine that it's a visual landmark. So I had to calculate the maximum visibility of every point of topography across the Balearic Islands. I use a resolution of 50 metres for this. If anyone has a better laptop that won't blow up, I could take it a bit shorter, but mine could not handle it. And essentially, I calculated this using a funky package in R called Geosphere, and a function which basically takes the curvature of the Earth and the elevation of any point of land, and using some Pythagorean theory and reflection from the Earth's surface, basically tells you at what maximum point that landmark would be visible. This is assuming that the bird is at sea level. Oh. So using the schematic here, we can see, so say the red radius is the radius of visibility of this mountain. For my analysis, I would say that the bird, while traveling along the blue line, is in absence of landmarks, and in red, it is in potential presence of landmarks. Now, to touch back on what I said earlier, it's really interesting that there's no land between where they forage and their island for this study, because it means that the data is split really nicely between portions where they had no landmarks available and then this more local scale where they do. And that is how I then carried on with this analysis. And I found that when testing these, that the homing precision of these birds is greater when landmarks are available, which I think is pretty cool. So first we'll look at instantaneous deflection, and this was the angular difference between their direction and the most optimum direction home. And we can see that when landmarks are present, the instantaneous deflection of these birds significantly reduces. And similarly, in homing efficiency, which was sort of their straightness along this beeline route, we can see that when landmarks are present, this homing efficiency significantly increases. The really important thing to note here, though, is that if you look at the x-axis, they are really small on this, so I'll tell you. Say we take homing efficiency. When landmarks are absent, the homing efficiency of these birds is still 0.95. Now that means that, again, if we go back to that Microsoft clip art example, that if the bird was traveling 100 meters, 95 of that, or approximately, is along its most optimum route. So they're already really good at what they do. And we're looking at really nuanced changes here in behavioral and navigational strategies when I'm looking at this landmark availability. So if we touch upon what I said earlier, these birds sort of use really wide-ranging spatial scales when they navigate. And that's where we see all these magnetic and other cues that could potentially be used. But I wanted to see that if at local cues, there is potentially different strategies. And I believe that's what I found. When landmarks are visible, homing precision increases. Now, that's not to say that they don't use the same cues that they did before, but something in this local scale is giving them better precision than what they had previously. But obviously, I'm a very keen first-year student, and I want to take this a lot further. I've got a bit of time to do it. So I want to try and identify some sort of decision points in these natural contrasts. So I think it'd be really interesting to see, specifically on this GPS track and on this bird's movements, when does its homing efficiency and its navigation improve? At what stage? Is it perhaps when the very first point of land comes into view? Or perhaps do they triangulate their position off of several landmarks? Due to the data and the resolution of tracks we have yet, we don't know. But I think with higher resolution data, so we get more of an insight into their movement, and perhaps some more advanced computational modelling that I don't know how to do yet, we may reveal these decision points. I've just had we had a really good tracking campaign on SCOMA this year, and we got some really, like a big push for higher frequency data. So I think that the potential answers to these questions might be on the horizon. So with that, I want to round up and say thank you. Thank you to all of my lab and my supervisors and everyone. And since I had the pleasure of being the last talk of the conference, I just want to say a big thank you to the organisers and volunteers, catering and everyone. You know, it's been an amazing conference, an amazing introduction to the seabird world. So thank you.
Thank you very much. I think as well we all agree with your sentiments there at the end.
Definitely.
That was brilliant. Is there any questions? Oh, there you
Several.
go.
Hey, really interesting talk. Thank you. I may have missed it, but how did you define whether landmarks were available or not? Like, did you use visibility?
Oh, yeah, sorry. So I think I sort of skipped over that. So the way that I would define that is where, so once I have this maximum visibility distance calculated, let's say it's, 100 kilometers, any GPS point that was within that range, so say the distance from that landmark to that bird was 95, I'd assume that this landmark is in theory visible to the bird. I'd like to sort of take the models further and sort of look at like field of view and things to see if sort of based on the direction of travel, is that landmark really visible to the bird? But I think that's going to be some more computational things down the line.
And that might obviously differ with weather and stuff as well.
Yeah, exactly. So one of the things I did sort of behind the scenes was I cut anything out of night, although they don't tend to travel at night. But anything sort of around sunset I was very aware of, just in case the visibility was reduced. Generally across the tracks, the weather was pretty good for them, or at least based on historical data, so I didn't include it this time around. But it's definitely, yeah, something to think about.
Okay. Thank you for a really interesting talk. I was wondering if you have considered marine landmarks, or like marine features that are stable in time. I mean, any colleague that ever crossed the polar front in the Southern Oceans knows when they cross the polar front in the Southern Ocean, even though there is no land around. So I'm not familiar with the Mediterranean, but are there any features, river plumes, anything other like seasonal chlorophyll blooms, something that they can actually see in the water that could be basically a stationary landmark?
Oh, no, that is really interesting. I haven't actually heard of marine landmarks before, so that's something I should definitely have a chat and include. I imagine for the smaller scale, since it's a sea, but perhaps not. But as you said, blooms and things are quite common in that area, so there could well be something that during the time they're there on the island, there's something giving them some local information. Assumably as well that it's because it's a familiar area, there might be really small things that they're picking up on that we can't see.
Cool. I think we've got a question at the back.
Yeah, one at the back. It's a race. Oh.
Hello. Awesome talk. I think we can all agree this is super interesting. I was just wondering, it's maybe not relevant to your study species or the Mediterranean just now or where you're studying, but do you have any thoughts on like novel new man-made structures that are getting put into the marine environment and how that might almost surprise, confuse birds? There's these new landmarks popping up. There's offshore wind farm turbines that they've relied on, and then they're coming back the year later and there's a turbine, or there's a ship that's moored for 3 weeks and then it disappears. If there's been any studies into whether this might affect birds and using landmarks for navigation in some way?
Yeah, that'd be really interesting. I mean, I don't think I'll say that we should put wind turbines near the critically endangered seabirds, but that would propose sort of a really cool, like, big experiment that you could see if we had GPS before and after, say, the installation of turbines, or like you said, boats, whether that could be something that they use. Maybe I imagine to begin with it would startle them, but you could also, it's in a sense putting a point of elevation in the middle of there. So maybe I'd see these effects have improved earlier in the track if there was landmarks like that around. There hasn't been much study, I don't think, of things of wind turbines are relatively new. Quite a few of us have done sort of pilot studies or experimented with landmark use sort of at a much smaller scale on like the colony and things, but we haven't found anything sort of concrete yet. I think the birds are quite clever at using, as I mentioned at the start, like a multitude of cues. And I think in their visual field, if they see something that's sort of off or they don't recognize, I mean, I can't speak shearwater, but I'd imagine that they kind of think, well, that's strange, but I know that all these other cues are the same, so I'll carry on using those instead. So, yeah.
Okay, great, thanks. I think we'll probably wrap up there. So I'm sure if you have any other questions for either Lewis or any of our speakers, they'll be hanging around for a while. So if we could just thank Lewis and all of our speakers again, please.